Hi everyone, welcome to my channel Srinivasa Biology Tutorial. For this section to discuss NCERT line to line chapter Cell, the Unit of Life, part Mitochondria. Next one is what? Mitochondria. See here mitochondria, the singular you call as a mitochondria. Here, unless the specifically the stained is not the easily the visible under electron microscopes. You should actually observe as a nucleus that mitochondria is not visible. Now, the number of mitochondria per cell, the variable, the depending upon the physiological activity of the cell. So, that mitochondria is present as in a different size and shape depending upon that cell activity. Now, in the term of the shape and size, the considerable as in a degree of variable, the variability of the observation. Now, you see as basically one particular the mitochondria, the typical mitochondria appear as in a sausage shaped or nothing but as in a cylindrical shape. They appear as in a sausage or they are cylindrical shape. The diameter of around as in 0.22 as in a 1.0 micromeurans average and 0.25 micromeurans and length around as what 1.0 to 4.1 micromeurans is present. Now, you see as in a one particular mitochondria, it is in double membrane structure is present. The mitochondria is in a the double membrane structure. See, in this mitochondria, the outer membrane is present. Say for example, I am taking as in one mitochondria, it is in a double membrane structure. It is in a double membrane structure. Now, the outer membrane and inner membrane is divided into as in a two laminal to distinctive to aqueous the compartments is present. So, here it is the outermost layer, it is in a their outermost layer, it is in a outermost layer. So, between these two layers, they are having as a fluid is present. This fluid is called as in a the perimitochondrial space, that is in a perimitochondrial space. Now, see basically here the inner membrane, here the inner membrane here the inner membrane to get form as some infoldings, the inner membrane to get produced as in some infoldings like, okay. Here the outer compartment and inner compartments they given like here the inner compartments they filled with as in the dense homogeneous substances is called as what matrix. So inside they're filled with as in a some matrix is present here inside they having as what completely in some matrix is present here as well as here some has in a DNA and then ribosome. So okay, like this organelles as present here. So this is simply called as a matrix. Okay, now here the outer membrane of the knee continues the limiting boundaries of the organelles as present. The inner membrane to form as a membrane, the infoldings is called as a cristae. So infoldings are called as what? The infoldings are called as a cristae. So these are called as a cristae. These are called as what? Cristae. Now, in this outside of the matrix, the cristae, the increase the surface of area, the two membranes are present to own the specific the enzyme associated with as a mitochondrial the function, the mitochondria, the site, the aerobic respiration. So in this mitochondria is present, it is the mitochondria to get help as what? In the respiration, the aerobic respiration purpose in this mitochondria to get helps. Okay, the energy of the innate and they produce as a cellular energy from as an ATP. Hence, this is called as a powerhouse of cell. So when this entire in this inside they having as an that complete cristae has any that some ribosomes as present. Their synthesis of as an ATP. The synthesis of what? ATPs. To synthesis of any ATP. So hence it is called as a powerhouse of cell. It is called as a powerhouse of cell. Now, they produce a cellular respiration from as in the ATP, hence it is called as in a powerhouse of cell. Now, the matrix also they possess as in a single circular DNA molecule. See here, I mentioned here single circular DNA also they are present, and few RNA molecules, ribosomes, 70s ribosomes as present, compartment required for as in synthesis of any proteins, and that mitochondrial to divide as in a fusion. Okay, the remaining to discuss in next class. Okay. Plastids. Plastids are found in all plants and euglenoids. These are easily observed under electron microscope or large. They are bearsome as a specific pigments, thus they are imparting the specific colors to the plants. So when the pigments is present, their plants to appear as in different colors. Based on their type of pigments, plastids can be classified as in a chloroplast, chromoplast, and then leucoplast. So the pigmentation based on the chloroplast, they are completely plastids are how many types? 3 types. One is in a chloroplast, second one is chromoplast, third one is in a leucoplast. First, the chloroplast contain as in a chlorophyll, the chlorophyll and carotenoid pigments, which is the responsible for the trapping the light energy essential for the photosynthesis. So mostly the light energy to get up, they are uptaking for the chlorophyll pigments as well as any carotene pigments. In this, the chromatophores, they are fat soluble carotenoids pigments like as any carotene and xanthophylls and other pigments also present. Here other pigment also they are present on the in a chromatophores. This used as part of the in a plant yellow color, orange and red colors. Okay. Now here the leucoplastids, they are in a colorless plastids. They are varied the shape of the size. They are stored as a nutrients. They are amyloplast and the eleoplast. Amyloplast stored as in a carbohydrates like as in a starch. Example potato. Eleoplast stored as in a oils and fats. Whereas in a the alleroplast stored as in a proteins. They stored as in what? Proteins. Now the majority of the chloroplast, they given as a one chloroplast diagram. In this chloroplast, majority of the chloroplast of the green plants are found in this mesophyll cells of the leaves. So if we take as in one particular leaf contains a mesophyll cell, in the mesophyll cell the chloroplast is present. There are the lens shaped or oval or spherical, discoidal, even the ribbon like within the width around as a 2 to 4. This is very important. 2 to 4 microneurons. The number of various per as in a 1 per cell, the completely chlamydomonas. In this chlamydomonas, each cell they having as in only one chloroplast is present. In a green alga, 20 to 40% of the cell has in a mesophyll cell. Now, this mitochondria chloroplast is a double membrane bond, and two inner chloroplast membrane is in a the relatively less permeable, and the space is limited by as in that inner membrane. The chloroplast is called as in stroma. So when you take as enter the chloroplast, inside they have as a fluid is present, that is called as what? Stroma. Now here the space they limited by as in a number of the organized the flattened membranous sacs are called as thylakoids. See here they are arranged as some stalk are arranged. These are called as what? Thylakoids. These are called as in a thylakoids. So these are called completely in a thylakoids. Now as in these thylakoids is present where they are present on the stroma region. The presence of the completely in that stroma part. Now they are completely they are organized as in the flattened membranous stalks are called thylakoid is present on the stroma region. The thylakoids arrange as stalks, a cone coil is called as an grana or grana of thylakoids, or the integral thylakoids. These are called as what? They are thylakoids also. In addition, there are in a flat, a membranous, the tubules are called as in a stroma lamellae. Between these two complete two connected, these are called as what? Stroma lamellae. Now here mostly the stroma is in a chloroplast consists of enzymatic required for that is synthesis of carbohydrates and proteins. Here inside they having as what? Complete a stroma is present. Am I? In the stroma the major function is what? It is to synthesize completely to get have a number of enzymes as present. Their carbohydrates and proteins are easily to get synthesized. And it is consist of a small the double stranded circular DNA molecule ribosomes also present. Inside they having as in a the DNA as well as in a one the ribosomes as present. Now these chlorophyll pigments are as in a present in the thylakoids. The chlorophyll pigments mostly present as where? On the completely thylakoid membrane. The ribosomes their chloroplasts are in a small 70s type of ribosomes as present. Here mostly they having as in a 70s type of or as in a 80s type of ribosomes also they are present here. Okay, that is next. Next, ribosomes. Here next, ribosomes. The ribosomes are the in a granular structures, the first observed under electron microscope. Dense particles they are observed as in a George Pedale in 1953. The scientist was George Pedale. They are noticed. They are the comprised the ribonucleic acid, RNA, and proteins, and are not surrounded by any membrane. So ribosomes are not surrounded by any membrane. Okay. Now, in that eukaryotic cells, ribosomes are mostly in a 8S type of ribosomes. They are having as an 8S type of ribosomes as present. Here 8S type of ribosomes. Here, while their prokaryotic cells has in a 70S ribosome. So see, mostly the prokaryotes is in a 70S, and then eukaryotes is 8S type of ribosomes as present. Now, each ribosome has in it two subunits as present. One is a large and the one is in a small subunit. So one is the large one and the one is in a small subunit is present. Okay. Now, the two subunits of 8S ribosome, one is the 60S ribosome and the one is in a 40S ribosome. One is a 60S and the one is in a 70S. And it is in a eukaryotes. And prokaryotes, one is complete in a 50S and the one is in a 30S. And the one is what? 30S. Now, S stands for as what? Svedberg unit. S stands for as what? Svedberg unit for the sedimentation coefficient. Now, it is in a indirectly that measured with as a dense and the size. The both 70S and 80S ribosome compressed as a two subunits. When the two subunits get connected, to automatically to get formed as a ribosome structure. Next, the cytoskeleton. An elaborate network filamentous proteinous structure consists of microtubules and microfilaments, and the intermediate filaments as present on the cytoplasm. It is refers to as what? Cytoskeleton. The inside of the entire the fluid they having some microtubules, some microfilaments, and intermediate filaments also they are present. Now, in the cytoskeleton in the cell involved in many functions such as give as mechanical support, motility, and maintenance of the shape of the cell. It is the major functions. What is that? First is mechanical support, second one is motility, and third one is maintenance of the cell of the shape and size. Okay, this is in completely cytoskeleton. Okay, now next the cilia and flagella. Next the cilia and then flagella. See that cilia flagella. In that cilia and flagella, it is a hair-like outgrowth of the cell membrane. They are completely outgrowth structures. The cilia is small structure, work as they are causing that movementation of either the cell or its surrounding fluid. So it is mostly to get helper as a movementation purpose. The cilia is help. And flagella is they are comparatively as a longer and responsible for as a cell movement. It is also in a longer, the thread-like structure. It is also help for as to get motility or movementation. Now, the prokaryotic bacteria also consist of as a flagella, but they are structurally different from as that eukaryotic flagella. So here, prokaryotes cells also the euglena is their flagella is present, but eukaryotes also they having as a flagella, but these both are different. Now, once you talk about as the flagella, you see the under electron microscope of cilium and flagella, they show as covered with as a plasma membrane. You see as that enter the structures to get appear as what? Then completely they covered with as one membrane, plasma membrane is present. It is a core is called as an axoneme. The center of the core is called as what? Axoneme. This part, the total the center part is called as an axoneme. The possess a number of microtubules running parallel to axis. The axoneme usually as a 9 doublets or radially arranged as in peripheral microtubules, and the pair is centrally located as in microtubules such as an arrangement of axoneme microtubules to get referred to as in 9+2 arrangement. See, here to get complete arranged as in 9 peripheral tubules. Like they get arranged as in 9 tubules. They completely to get arranged. They get appear as in a 9+2 arrangement. To get arranged as in a 9+2 arrangements. Okay. Now, the central tubules are connected with as in a bridge, or also called enclosed as in a central sheath, which is connected with as in a one tubelet of each peripheral tubule the radial spoke. So it is complete to get arranged as in a spoke-like structure. It is arranged as in a radial spoke-like structure. It is in a peripheral tubules. These are in a peripheral tubules. Peripheral tubules. Okay. Thus there are a 9 radial the spokes as present. They are interconnected with as in linkers. They are both cilia and flagella emerge form as in a centriole. It is look like as in a the basal bodies. So it is in a completely in centriole like structure. So this it is called as in what? Basal body. It is called as what? In a basal body. Okay. This is in a completely what? In that sense the cilia and then flagella. Next the centrosomes or centrioles. Centrosomes and centrioles. Now see, in the centrioles, the centrosomes are organelles usually containing the 2 cylindrical structure. They are called as in centrioles. So there is the cylindrical structures. See, these are the cylindrical like structures. They noticed on the cytoplasm. Now they are surrounded as the amorphous. Their pericentrial nuclear material is present. The both centrioles, the centrosomes are perpendicular. The perpendicular each and other is also organized look like as in a cat wheel. So it is to complete to get appear as in a one cat wheel like structure. It is to get appear as in what? One cat wheel like structure. And in this entire the centrioles to get arranged as in a here also they are like to get arranged as it is a central core part. It is called as a hub region. It is a hub region. Here also 9 spoke fibers to get arranged as in a parallelly. It is to get also arranged as in a completely parallelly. Like they get arranged as in a in completely 9. Like these are the triplets. These are the triplets. These are the complete what? Triplets. Now they are made up of 9 evenly spaced as in peripheral fibers, the tubulin protein. So these are completely they are made up of what? They are completely made up of as in a tubulin protein. This completely they are made up of what? This total 9 spoke fibers they are made up of as in a they are tubulin protein. Now each peripheral they are fibril triplets. They get arranged as what? Triplets. 3 triplets as present. Now the adjustment of the triplets also they are linked. So see here this is to everyone they are linked. It is called as where the center of the region is called as in a hub region, which is connected with as in a tubules. The peripheral the triplets the radial spokes. It is made up of as in a protein. So this total protein fibers of the spokes to get arranged as in a to connected with as in a hub region. Okay. Now this centrioles form as in a basal body, cilia and flagella in the spindle fibers that gives rise to as in a the spindle. The apparatus during as in a that division of in cells in animals. In that animal cell, they are mostly to get help for as what? Cell division. Here centrioles the major function they help for as in a their cell division in animals. Okay. Next. Like, share and subscribe. Thank you for watching.